Hello, this is Alice Shapiro, your instructor for HO 150, Human Body Systems 1. This is the PowerPoint recording for the section chapter on blood. So first of all, some of the characteristics of blood. Blood is a type of connective tissue. It is sticky or viscous. It is heavier than water, and the amount of oxygen determines the color of blood. The more the oxygen content, the brighter red the blood will be. Blood is not really blue, but it will be less red the less oxygen content, or less bright red. The temperature is slightly higher than the rest of the body. Males on the average have 5 to 6 liters of blood. Females on the average have 4 to 5 liters of blood. And here is our famous model here showing us the contents of blood, the formed elements, which are the solid materials of blood, the cellular materials of blood, comprise about 45%. Plasma, the liquid portion, comprises about 55%. And this shows you the percentage by weight of the different substances dissolved in the plasma, and also what the formed elements are. You don't have to memorize these, but please familiarize yourself with them. We are going to be taking a look at the functions of some of these proteins and so forth, and the functions of the cells, including the white blood cells as well as the red blood cells. This tells you the composition of blood. Plasma is 55% of the total volume, and this tells you the characteristics of the plasma and also the formed elements. What is contained in the formed elements are cells and cell fragments. The platelets are actually cell fragments. The types of formed elements are erythrocytes, which literally means red cells, leukocytes, which literally means white cells, and thrombocytes, which actually means particle cells. Plasma proteins, this tells you what percentage and what the main functions are for each of the plasma proteins. I advise you to review this. And here we go with erythrocytes. Here is a wonderful example, I think, of the relationship between structure and function. What is the function of the red blood cell, the erythrocyte? To transport oxygen, O2, to the tissues. And the structure really facilitates that function. If you notice what we call this a biconcave disc, which means that it is concave both, let's say, at the top and the bottom. Think about this in terms of surface area. This increases the surface area of this red blood cell to allow for the heme portion of the hemoglobin, the iron portion, to attach to and release oxygen. Also, the contents of the red blood cell have been modified. Red blood cells, mature erythrocytes, do not contain a nucleus. They contain no organelles. Basically, they contain hemoglobin molecules, so they are, we could say, streamlined and specialized to carry oxygen. A red blood cell will live, I should say, function for about 120 days, and then we'll see later on how its contents are recycled and reutilized. Hemoglobin is the main component of the erythrocytes, as I have said, and its function is to transport oxygen. This tells you a little bit more about the molecule. The oxygen actually binds to the iron portion of the hemoglobin, and when O2 is attached, it is called oxyhemoglobin. Erythrocytes are stimulated by a hormone called erythropoietin, which is produced actually in the kidneys. The kidneys detect decreased blood oxygen, and this increases the production of erythropoietin, Erythropoietin stimulates red bone marrow to produce more erythrocytes. Increased erythrocytes, of course, cause an increase in blood oxygen levels, and therefore we have another negative feedback system going on. When O2 levels fall, more erythropoietin, more erythrocytes, more oxygen transported in the tissues, oxygen levels go up, erythropoietin goes down, negative feedback. And here is a diagram showing you decreased blood oxygen is recognized by the kidneys. They produce erythropoietin, stimulates red bone marrow, the stem cells in the red bone marrow, to produce mature erythrocytes that increases blood oxygen, and the negative feedback occurs. Fate of old erythrocytes: they are recycled. They're removed from the blood by macrophages in the spleen and the liver. The hemoglobin is broken down. The globin, which is a protein, is broken down into its component amino acids, which are then reutilized in other protein synthesis. The hemoglobin's iron is recycled. Heme itself is converted to bilirubin, and the bilirubin is taken up by the liver and released into the small intestines as part of the bile. So we have in our own bodies many examples, and this is a really good one, of recycling and reusing. So we could say, perhaps to sort of have a little pun here, that we have green going on in our bodies. Here, this is diagrammatically the hemoglobin breakdown. You can see this diagram in your book. It shows you via pictures what I have just explained to you. Here is a macrophage from the spleen and liver starting the recycle, reusing process, and all of the organs here that are involved. Leukocytes are white blood cells, abbreviated WBC. They lack hemoglobin, therefore they are white. They're larger than erythrocytes. They contain a nucleus, which means they can reproduce themselves. They function in the immune system and also in phagocytosis. There are several types of leukocytes. Please review the types and understand each one of their basic functions. Here are the first three: neutrophils, eosinophils, basophils, then macrophages, and lymphocytes. Platelets are really cellular fragments. They are involved in blood clotting, and they are also produced in red bone marrow. And this shows you the relative proportion in a smear of blood of platelets, which are much smaller, erythrocytes, which are the most numerous, and then a couple of kinds of white blood cells, which you can see are much larger and do contain, each one contains nucleus. Hematopoiesis means the process of red blood cell formation. In an infant, it occurs in the liver, thymus gland, spleen, lymph nodes, and red bone marrow, and in adults, mainly in the red bone marrow. And the original cell line is a stem cell, which is a precursor to the ultimate formation of all of the types of blood cells: white blood cells, platelets, which you can see are cell fragments of a megakaryocyte. You don't need to memorize this process, but I think it's interesting and informative to take a look at this diagrammatically. So stem cells, all of our blood cells and our platelets, white blood cells, red blood cells, originate from a primordial stem cell, which then differentiates to fulfill the various needs and functions. All right, let's take a look at blood reactions, negative reactions that can occur. Transfusion reactions, or agglutination, is the clumping of blood cells. This is bad. If you have a blood transfusion, you do not want agglutination. Why? Because that will clog your vessels and can lead to cellular destruction and even death. So you want to make sure that there is cross-matching before you receive a transfusion. Antigens are the molecules on the surface of red blood cells as well as other cells. Antibodies are proteins in the plasma, and these two can interact and cause adverse reactions. Blood groups are named according to a certain antigen. There are A antigens or B antigens, or in the case of type O blood, none of these antigens on the surface of red blood cells. Here are the ABO blood groups. Blood type A has antigen A. It will produce antibodies against B. So if you have type B blood and you are type A, your blood will agglutinate. So you do not want that. If you are type B blood and you have type A antigen given to you via transfusion, it will agglutinate. Also not a good thing. If you are type AB, you have both the antigens and you do not have the antibodies. So you are called the universal recipient. You can receive either the A antigen or the B antigen or both, and no negative reaction in this regard will occur. Type O blood do not have these antigens at all, so they produce antibodies against A and B, so they cannot receive blood from any other type but type O, but they are called the universal recipient because they can donate blood to A, B, or AB. Please review that. This is diagrammatically what the adverse reaction may look like, and showing you that it's kind of a geometric, like all cellular receptors, it is a geometric relationship. If you have antigen A, you have a certain receptor that has a certain geometry. If you're antigen B, you have a certain geometry. If you are type AB blood, you have both antigens and you do not produce antibodies against A or B. Take a look at this diagram. I think it will help to clarify, showing you on the next page what will happen if you are type A blood of a donor and you are also type A blood. You will not produce antigens against this type. You will not produce antibodies against this type. The antibodies you do produce will not cause agglutination because they have a different geometry. If you receive blood from a B recipient, you do have the antibodies that are the correct geometry to agglutinate with the type B blood, and you will have an adverse reaction. And this explains what I said before. O are the universal donors because they do not have these antigens. Type A can receive blood from either A or O, and so forth. If you don't understand this concept, please study and message to me to clarify this for you. It will also help in understanding other tissue rejection reactions because they are all similar to an adverse reaction to blood transfusion. Rh blood is another antigen. Rh positive means you have the Rh antigen and you are among the 85 to 95% of the population who has this. Antibodies can only develop if an Rh negative person is exposed to Rh positive blood either by transfusion, as before, similar to A and B reactions, or from mother to fetus. So if you are an Rh negative woman going to have a baby, you better check and see whether there is a chance that your child will be Rh positive. If the father is Rh positive, then there is a good chance the baby could be Rh positive. And the first time, because it takes some time for antibodies to form, the mother's blood will produce antibodies, but will probably not adversely affect the fetus or the child being born. But in subsequent births, the immune system has a memory, and so this reaction can occur very quickly and cause an adverse reaction. And it can cause hemolytic disease of the newborn, which could be fatal to the fetus. There's a drug called Rogam, which contains antibodies against the Rh antigen, which can be injected into the mother to help to alleviate this response. But here, diagrammatically, is what can happen. Please review this diagram. It is from your textbook so that you will understand. Remember, the immune system, and this is really part of an immune reaction, does have a memory, so the first time adverse effects will probably not occur because there isn't time for this reaction. Subsequent births are much more risky. All right, I'm finished with the blood part of the PowerPoint narration, so please contact me if you have questions.